in evolution. He was a student with Dr. Leo Buss and Stephen de la Potra, and using the cnidarian Hydractinia as a model to understand the molecular determinants of allorecognition. As a postdoc, he went to Harvard and worked with Dr. Strominger, who's a classical immunology guy, and in this perspective, he was studying molecular determinants of non-self recognition from a vertebrate perspective. And then he took on a job at Pitt, and he's an assistant professor now, and he's exclusively back 100% to his Hydractinia passion. And I'll let him tell you the rest from here. After the talk, we're going to kind of mosey on over to the tavern and hang out there for a while. And if anybody who wants to join us, we'll make a decision on where to go eat. We'll go have some dinner. You guys are welcome to come.
Thank you, Larry. You made that sound a lot more sordid than it actually is. Can you all hear me? Yes? Okay, good. I'm getting over a cold. I don't normally sound this close to Barry White, but that's where we are. If you wouldn't mind hitting the lights. So I'm going to talk to you about Hydractinia allorecognition. And since most of you, I'm sure, don't think about either of those two words very much, I'm going to spend quite a bit of time at the beginning going over the basic sort of system and what we study so that we can all be on the same page. So allorecognition, first off, this is Hydractinia. This is the organism that we study. We'll tell you more about that as we go. Allorecognition can be basically defined as the ability to distinguish between your own tissues and those of other members of the same species. And so I am in a medical school, in a transplant institute, and so of course immediately people think about transplant rejection as an example of this type of thing. But it turns out that a lot of colonial invertebrates like Hydractinia also do something called allorecognition, but under natural circumstances. So I'll go over sort of what the model system is, and then some mostly published work about the molecular basis of how Hydractinia makes this distinction between self and non-self, and then at the end talk about what we're doing now to understand how they actually make that discrimination at a functional level. So Hydractinia is a cnidarian, so it's related to jellyfish and anemones and corals. Hydractinia does not live in these beautiful environments. It lives on the back of hermit crab shells, and the place where we collect them is Long Island Sound, which is, has a visibility of about 1 foot. But this is what it looks like sort of in situ. It's this fuzzy thing on the back of the hermit crab. The common name is actually spiny snail's fur, despite the fact that this is obviously a crab and not a snail. And in the lab, of course, we don't grow it on the crabs because they are a real pain in the butt to keep, and so we put them on microscope slides and grow them in 10-gallon aquaria. And this is an example of how we do that. And basically the way you do this is you basically just cut a little piece of the colony, and since they can reproduce asexually, they'll attach to that surface and grow, and then you can propagate the same clone over multiple slides. And in the lab, you can keep them for years, genotype for years. So the longest that we've ever had a single genotype has been about a decade. And then it got sick and died. I don't know if that was because it got old or if it just got sick. But if you were to zoom in on one, this is what it would look like on the slide, and then this is just a schematic. And of course, every one of these parts has a fancy name, but the basic 4 parts you need to know are each colony has polyps. These are sort of the feeding structures, just like in a coral. So they catch food. In the lab, we feed them brine shrimp. There's a mouth at the center of this whorl of tentacles. They'll digest the food in that body column, and then they'll actually pump that food to other polyps and the rest of the colony through this network of gastrovascular canals. These are basically just tubes that are embedded in this plate of tissue, which is called the mat. And sometimes those tubes extend outside the mat, and we call those stolons. So that's the basic 4 parts of a Hydractinia colony. So like all cnidarians, Hydractinia is a diploblast, so it doesn't have any organized tissues. It basically has a gastrodermis. This is a picture of a hydra, but it shows the histology fairly nicely. There's an epidermis, a gastrodermis, which lines the gut, And then in between, there's a thin layer of mesoglia. That's the jelly in jellyfish. All cnidarians have that, separating the cell lineages. And Hydractinia just takes this form and iterates it over a colony. And they do have some specialized structures. So, for example, they have feeding structures, and they also have reproductive polyps. So the colonies are either male or female. Colonies, either one or the other. And the males, here's the males here. They have these gonophores here that are loaded up with sperm. The females have eggs. And what they do is if you keep them on a day-night cycle, you turn on the lights, an hour later they release eggs and sperm into the water. You can collect that and actually breed them in the lab. And they will fertilize and over a couple of days form a crawling larva. You can force that larva to metamorphose with some chemicals. It turns into a primary polyp, and then by asexual budding, it buds off some stolons and then makes some more polyps, and eventually you end up with a mature colony. And they can complete this in about 3 months, which is about the same span as zebrafish, which is a model system that a lot of people know about in terms of aquatic model systems. So what we study is what happens when two colonies come into contact with each other. And so this is just, these are colonies that we grew by basically taking small pieces of adult colonies and putting them next to each other on the same slide. So Larry, I said, is going to point out the chitin. That's this orange stuff here on the bottom here. And so these maybe have been growing for about 4 or 5 days. And over time, they're going to grow into contact with each other. And if they are incompatible, which is most of the time if you took two colonies off of different shells and put them next to each other, they would be incompatible with each other. And they would start to do something called rejection. And so if we zoom in here, what you can see is that they have built up this border. And there are actually these stolons that are starting to grow from one colony on top of the other colony. These are stolons that differentiate in response to incompatible colonies. They lift up off the surface, and they start growing over the opposing colony. And if you were to zoom in even more under light microscope, these are old pictures from a paper that I was not part of, but they show it nicely. What you would see is that the tip of that growing stolon is loaded up with these spheres here, or these oval structures. These are the nematocytes, so the stinging cells that all cnidarians have. They're like little harpoons, if you've ever looked at one or seen an illustration of one under a microscope. And the way they typically work is when they're triggered, they pierce the integument. This is like an inverted rubber glove that gets exuded into the prey item usually, and then a neurotoxin gets shot out the end, and that's how they paralyze their prey and catch it. In this case, the growing stolons are actually loaded up with these nematocysts, and then when they encounter an incompatible colony, they form a battery opposing the opposite colony, and that colony that just got contacted, here there's not really any organized structure of those nematocysts. Here, it also forms a battery of those nematocysts. These are cells that are recruited into the area, and they orient it to the opposing colony. And then at some point, there's a trigger, and they both fire off. And then subsequently, those stinging cells will migrate into the area, flip their nematocysts around, and fire them off at each other. And so they'll keep doing this and doing this and doing this until they destroy whatever tissue's around. So if you were to zoom out a little bit now, this is a SEM, a scanning electron micrograph, of a rejection that's going on, and this is the end of one stolon here that's attacking one of these polyps, and all these little threads are those nematocysts that have fired off at the polyp, and that will eventually destroy the polyp. And so if we come back to our picture of a rejection, this is an early rejection. If you come back in in about 2 or 3 weeks, the other colony is completely gone. This colony has won the fight, and it will then monopolize the substrate. So it's not always as violent as this. If you have compatible colonies, and these are usually colonies that are closely related or pieces of the same colony, when they come into contact, they're compatible and they'll actually fuse together. And so if we were to zoom in here, this is the original explants. Now they're growing. You can see there's these little extensions here. Those are stolons that had contacted. And if we had caught it, what we would have seen is the tip of one stolon touching the side of the new colony. They both, again, make those batteries of nematocysts that are oriented towards each other, but instead of fighting, they just go away. And within about 2 to 4 hours, they establish a common lumen and a single gastrovascular system. Okay? And so that's what happens here. These are actually, from this point on, physiologically integrated, and they'll continue to grow and become one larger colony until, if you didn't know any better, you would come and look at this and you'd just say it's always been one colony. Now there's a third intermediate response that sometimes happens, which we call transitory fusion. And in this case, what happens is the colonies will initially come into contact, and they'll fuse the way I just described. And if you were to zoom in here, you could actually see along this margin. This was 24 hours after they had contacted. These are the gastrovascular systems, or gastrovascular canals. And I hope you can see that they're open. And you could actually, if you fed one side, you could actually watch food go back and forth between them. But within the next 24 hours, that border separates, and they establish a clear border and basically a new edge. So they've gone from being physiologically integrated to separating their tissues and making a new edge. And this has nothing to do with nematocysts that I just told you about. It has to do with cell death. We don't really understand how. But the cells that are in this region die in a regulated way, and they reorganize their morphology to create a new edge. And there's no memory to this response, right? So if I were to cut these two colonies back, which you can do, they'll heal just fine. They'll go back into contact and do exactly the same thing over and over and over again. So we have fusion, transitory fusion, and rejection. Why? So why do they do this, right? This is really cool, but what's the purpose? So it has to do with two things mainly. One is intraspecific spatial competition, right? So remember, these guys are living on hermit crab shells. And because they're colonies, the larger they get, the more resistant they are to disease because they're less likely to be killed off by a small infection. They have a greater gametic output because they can make more reproductive structures. And so it's really to their advantage to monopolize that surface. And so they want to fight off every other colony that's on that shell, but they want to make sure that as they grow around a shell, right, it's a three-dimensional structure, they're going to encounter themselves. They want to start fighting with themselves on the opposite side because that would be not a good idea. And it's actually worse than that for Hydractinia because Hydractinia starts out as a little larva that crawls onto the hermit crab shell. And they actually settle in very characteristic spots. So this is just looking not at one shell, but where larvae can be found out in nature as single polyps. You can see they like to sort of cluster in these ridges and along the apertures. And so from a very early start, they're encountering other Hydractinia and having to decide, is this me? Is this not me? Do I fight them off? And if they can win that fight, then they have a much better chance of taking over that shell. So it's really important ecologically from that perspective. But there's another thing about this that makes it really important that they don't make a mistake and accidentally fuse to a colony that's not closely related or actually in fact themselves. And that has to do with their stem cells. So Hydractinia is a hydroid. All hydroids have a stem cell lineage called an interstitial cell. And it's a stem cell lineage that, in this diagram here, can give rise to every other cell type in the organism. So gland cell, these are digestive cells, nerve cells, the nematocytes, but importantly the germ cells. Okay? And since these cells move around, if two colonies fuse, now suddenly those stem cells are moving back and forth between two colonies. And these stem cells will actually compete with each other for access to the germline. So you can imagine if a colony fuses to another colony, those stem cells come in, they start competing, and your body could end up making gametes of the other colony, right? And from an evolutionary perspective, your fitness plummets. And so it's really important to make sure that you're only fusing to self or at least closely related individuals to mitigate this threat of stem cell parasitism. And so this is a problem not just for Hydractinia, but most colonial sessile invertebrates, so things like sponges, other cnidarians like corals, bryozoans, and chordates. So this is an ascidian, Bacillus in particular. It's essentially your closest living invertebrate relative, or one of them. Have allorecognition systems to deal with this problem of having to make sure that they don't fuse to unrelated individuals. So one thing about these systems, for a long time, people have known about the existence of these systems, but nobody's really known whether there was any evolutionary relationship between the allorecognition systems that we see in sponges and cnidarians and bryozoans and sea squirts and the allorecognition responses in invertebrate immunity that we study in the Transplant Institute. And so a big question is whether there are any deep homologies between these systems. And that's one of the main things that my lab is trying to study, and the fact that we're in the Transplant Institute is why, or that's why we're in the Transplant Institute. Of course, if you want to answer those questions, you need to start understanding the molecular basis and what genes are involved. And there really, until recently, had been no information about that. So what I'm going to tell you about is the work that we've done working on one cnidarian to try to address that problem. There is one other system that I'm not going to talk about today, but you should be aware of. In Ascidians, there's a lab that's headed up by Tony DiTommaso at UC Santa Barbara. They also studied allorecognition and found it to be actually very interesting and completely separate story from the one I'm going to tell you today. And of course, the motivation that we use for studying this in the Transplant Institute is that perhaps if there is some evolutionary relationship here, perhaps we could use this to find clues to mechanisms of allorecognition and self-non-self recognition in the vertebrate immune system that we've overlooked because we haven't been looking in these particular regions or areas before. So, okay, enough talk. How does that actually work? Okay. So, as you might imagine, Hydractinia is not your classical model system, and so we've had to create a lot of the tools necessary to get at this question ourselves. And by ourselves, I mean not just myself, but my PhD advisor back before I started. And what he decided to do was to create some inbred lines of Hydractinia. Because you can breed them in the lab, this was possible. And so don't panic about this pedigree here. There's only two things you need to take away from it. The first is that he took two colonies out from nature. These colonies did not fuse to each other, but he bred them together. And then in every subsequent generation, the parents of the next generation were chosen such that they always fused to each other. Okay? And then at some point, another colony was brought in to create another line. And these lines were continued such that every colony within this lineage would fuse to itself, but would reject the opposite lineage. Okay? In a sense, what this is doing is it's whatever genes are controlling allorecognition, it's fixing each line for different alleles of those genes. Okay? And in our lab, we called them F and R. Just, I don't know why, that's just what they were called. So that's great. The second thing that was done was, in the same background, a near congenic line was created. And the purpose of this lineage was to take these R alleles, controlling allorecognition, and bring them back into the genetic background of this first lineage. Okay? What this essentially does is gives you an animal where, or a line where all the variation between individuals in this line is localized to the region of the genome that's controlling the trait that you're interested in. Okay? Now, once you have that, you've narrowed down your problem, and you can use that polymorphism to identify markers, essentially, bits of DNA that differ between the two phenotypes. And if you have that, you can actually do mapping experiments and localize it to a region of a chromosome, and that's what's done here. So I'm skipping over like 15 years of genetics here to get you to this point. And don't worry, the details aren't really important. What's important is what we discovered was that there are two genes called Alr1 and Alr2, and together these control allorecognition in Hydractinia. Okay? And the way they do that is as such. So it's essentially a matching game. Or immunologists like to give these things funny names, and so they call this missing self-recognition. And so what happens is if you have two colonies, and I'm just sort of drawing you cartoons of the chromosomes with different colored alleles. So here's colony number 1 and colony number 2. They share a red allele at both Alr1 and Alr2. If you share an allele at both genes, you fuse. Okay? If there's only one gene where there's a shared allele, so in this case, at Alr2 there are shared alleles, but at Alr1 there are no shared alleles, you get this transitory fusion response that I told you about. And if there's no matching alleles between the two genes, you end up with rejection. Right? And this sort of intuitively makes sense, right? Now, we went in and, again, through a lot of genetics I won't sort of go into the details of, by positional cloning, identified the actual genes that are responsible for this. So Alr1 and Alr2 were just locations on the chromosome. We didn't know what genes were there. But we figured out what genes are there, and the proteins that they encode for look like this. So Alr1 and Alr2 are both transmembrane proteins. So these are proteins that are sitting up on the cell surface. Okay? They have cytoplasmic tails, which do not have any predicted homology to any proteins that we know about. But their ectodomains are kind of interesting because they contain either 2 or 3 tandem domains, which if you look in the databases at NCBI for things that are homologous to them, what you find is that they're similar to immunoglobulin-like domains. So these are the same kinds of domains that you find in antibodies and in T-cell receptors, but also in lots of other proteins and animals that are involved in protein-protein interactions. So it's actually not that surprising you find Ig-like domains. And then in between these Ig-like domains, there is a region which we just call a spacer. It's a region where there's no predicted homology or structure. So these domains, they are similar to immunoglobulin-like domains, but they weren't obviously orthologous or homologous to particular genes. And so we were wondering, how likely it is that they actually fold like Ig-like domains. And so this looks fancy, but all we did was put the sequence into a computer algorithm and it spits out a model. But basically what the model shows you is that they're predicted to fold like an Ig-like domain. So an Ig-like domain has a characteristic fold. It looks like they call it a beta barrel. It looks like sort of a barrel. And both for ALR1, both of those domains, and for ALR2, all of these are predicted to have Ig-like domain folds to varying degrees of specificity. Now we're actually working to crystallize these things and figure out what the actual structure looks like, but for right now, that's where we are. But that's not even the most interesting part about this. The most interesting part is that they're very polymorphic. So when we first discovered these, we went out and picked a handful of colonies from nature and sequenced the alleles, right? So you expect to see 2 different alleles in each organism. And when you take those alleles, 20 alleles for ALR1 and 26 alleles for ALR2, and you align their sequences, and then you just look for each position and see how variable is that position between individuals. And if you plot that variation in a histogram like this, H prime is just a measure of that variation, what you see is that for ALR1, the ectodomain is extremely polymorphic. Nearly every site has variation in it. The spitoplasmic tail is relatively conserved. Again, for ALR2, the ectodomain is very polymorphic. It seems like this first domain is even more polymorphic than the rest of the region, and the cytoplasmic tail is very conserved. This is what you'd expect for a molecule that's involved in self-recognition, right? You want to have lots of variants out there because the more variants there are, the more precise your system's going to be. For ALR2, we did a proper population study. I'm saying we, a postdoc in the lab while I was there. I didn't actually do this stuff, but I'm going to show you the data. Went out and just in this one population here in Lyle and Sound, took 239 colonies, sequenced out of these animals 473 alleles. That's just the first couple exons, which encode just domain 1. So she didn't look at the whole protein, but just that first variable domain. And what she found was that there were 198 distinct nucleotide sequences and 183 distinct amino acid sequences. Okay? Normally, you would not find this many alleles at a single gene in a population like this. Okay? This is extremely polymorphic. So if you know anything about MHC